Hello everyone, myself Amrita Yadav, Assistant Professor, School of Pharmacy, BPTU. Today I am going to discuss about chromatography. Chromatography is originated from a Greek word. Chroma means color and graphic means writing. It was discovered by a Russian scientist whose name is Svet, which is referred as father of chromatography and is also credited for the development of chromatography. Now the definition: chromatography is a technique for separating mixture into their components to analyze, identify, purify and or quantify the mixture or components. The components to be separated are distributed between two phases, namely stationary phase and the mobile phase. Principle of the chromatography. Chromatography is usually based on the principle of partition and adsorption of solid between two phases. It mainly consists of mobile phase and a stationary phase. The mobile phase usually refers to the mixture of the substances to be separated, dissolved in a gas or a liquid. The stationary phase is a porous solid matrix through which the sample contained in the mobile phase percolates. Adsorption chromatography. Adsorption chromatography is one of the oldest type of chromatography which utilizes mobile liquid or gaseous phase that is adsorbed onto the surface of a stationary solid phase. The equilibration between the mobile and the stationary phase accounts for the separation of different solutes. In other words, we can say that adsorption chromatography is a process of separation of components in a mixture introduced into chromatography system, which is based on the relative difference in adsorption of components to stationary phase, which is present in the chromatographic column. The other one is partition chromatography. Chromatography in which separation is based mainly on differences between the solubility of the sample components in the stationary phase or on the differences between the solubility of the components in the mobile and the stationary phases. This form of chromatography is based on a thin film which is formed on the surface of a solid support by a liquid stationary phase. Solute equilibrates between the mobile phase and the stationary liquid. Classification. Chromatographic methods can be classified in three different ways based on shape of chromatographic beds. Example, planar and column chromatography. Based on the physical state of mobile and stationary phase. Example is gas and liquid chromatography. Based on mechanism of separation. Example, ion exchange chromatography, partition chromatography, affinity and adsorption chromatography. Classification based on shape of chromatographic beds. It consists of two parts, planar and column chromatography. In planar chromatography, stationary phase is present on a plane. The plane can be a paper impregnated by a substance acting as a stationary phase, for example paper chromatography, or a thin layer of a substance acting as a stationary phase spread on a glass, metal or plastic plate, for example thin layer chromatography. Planar chromatography is also known as open bed chromatography, for example paper chromatography. Coming to the paper chromatography is a liquid partition chromatography. In paper chromatography, the end of the paper is dipped in a solvent mixture consisting of aqueous and organic components. The solvents soak in paper by capillary action because of fibrous nature of paper. The aqueous component of the solvent binds to the cellulose paper and thereby forms a stationary phase with it. The organic component of the solvent binds continues migrating, thus forming the mobile phase. Mechanism of the separation. Mobile phase rises up by capillary action. S sample is concentrated as a minute spot at the bottom of the filter paper. After that, the sample mixture gradually rises up with the mobile phase, which is liquid. Compound in the mixture will be separated according to their ability of their solubility. More polar substances will move slower and less polar substances will travel faster. Procedure of paper chromatography. A small spot of the sample is applied to a strip of chromatographic paper about 2 cm away from the base of the plate. This sample is absorbed onto the paper and may form interaction with it. After that, the paper is then dipped into a solvent such as ethanol or water, taking care that the spot is above the surface of the solvent and placed in a sealed container. The solvent moves up the paper by capillary action and dissolves the sample mixture, which is then travel up the paper with the solvent solute sample. Different compounds in the sample mixture travel at different rates. It takes several minutes to several hours. Now coming to the analysis, spots corresponding to different compounds may be located by their color, UV light, ninhydrin, or by treatment with iodine vapors. Significance of paper chromatography: it is very simple, easy, rapid and highly efficient method of separation. It can be applied even in micrograms quantities of the sample. It can also be used for the separation of wide variety of material like amino acids, oligosaccharide, glycosides, purines and pyrimidines, steroid, vitamins and alkaloids. Second example is thin layer chromatography. In thin layer chromatography, stationary phase consists of a thin layer of adsorbent materials such as silica gel, ammonium oxide or cellulose immobilized onto a flat carrier sheet. A liquid phase consisting of the solution to be separated, which is dissolved in an appropriate solvent, is drawn up the plate through capillary action, separating the solution based on the polarity of the compounds. Significance of thin layer chromatography: it is used for the determination of plant pigments, detection of pesticides or insecticides in fruits, identify compounds present in a given substances, and finally monitoring organic reaction. Coming to column chromatography, in column chromatography the stationary bed is within the tube. In column chromatography, the stationary phase may be pure silica or polymer or may be coated onto, chemically bonded to or supported particles, depending on whether mobile phase is gas chromatography or liquid chromatography. When the stationary phase is liquid chromatography, it consists of a small diameter particles. The technique is called high performance liquid chromatography, that is HPLC. Another example is gas chromatography. Gas mobile phase is used to pass a mixture of volatile solutes through a column containing the stationary phase. The mobile phase, often referred to as a carrier gas, is typically an inert gas such as nitrogen, helium or argon. Solute separation is based on the relative differences in the solute vapor pressure and the interaction with the stationary phase. Thus, more volatile solutes elutes from the column. A solute that selectively interacts with the stationary phase elutes from the column after with lesser degree of interaction. The column effluents carries separated solutes to the detector in order of their elution. Solutes are identified qualitatively by their retention times. Peak size is proportional to the amount of solute detected and used to quantify it. Liquid chromatography. In liquid chromatography, separation by liquid chromatography is based on the distribution of the solutes between a liquid mobile phase and a stationary phase. When particles of small diameter are used as a stationary phase support, the technique is HPLC. This is the most widely used form of liquid chromatography. Classification based on phases. This depends on the nature of the mobile phase and the stationary phase. It is of two types, solid phase and the liquid phase. Example of solid phase is solid liquid chromatography, solid gas chromatography, and the example of liquid phase, liquid chromatography, liquid gas chromatography. Classification based on mechanism of separation. These include ion exchange, partition, adsorption, size exclusion, affinity, mechanism. Ion exchange chromatography is based on exchange of ions between a charged stationary surface and ions of opposite charge in mobile phase. Depending on the conditions, solutes are either cations positively charged or anions negatively charged. These are also known as ion exchangers. Ion exchangers are made up of two parts, an insoluble matrix and chemically bonded charge groups within and on the surface of the matrix. It is classified as cationic or anionic based on whether it exchanges cation or anions. Cation exchangers are also known as acidic ion exchanger. Anion exchangers also known as basic ion exchanger. Each type of exchangers is also classified as strong or weak depending on the ionizing strength of the functional group. Bound molecules can be eluted by altering the pH of the eluting buffer or by increasing the salt concentration of the eluting buffer. A positively charged protein bound to cation exchangers can be eluted by increasing the salt concentration in the eluting buffers because cation present in the buffer competes with a positively charged group on the protein for binding to the ion exchanger. Protein having a low density of net positive charge will tend to emerge first, followed by those having a higher charge density. Selection of an ion exchanger. The choice of ion exchanger for the purification of biomolecule largely depends on the isoelectric point of the biomolecule. A solute having a positive charge will bind to a cationic exchanger and vice versa. Many solutes having more than one type of ionizing group and may have both positively and negatively charged group. The net charge on such molecules depend on pH. At isoelectric point, the solute has no net charge and would bind to any type of ion exchanger. Choice of buffers. Cationic buffers should be used with anionic exchangers and vice versa. Firstly, the pH chosen for the buffer depends on the range of stability of macromolecule to be separated. Second, the buffer pH should be chosen so that the desired macromolecule will bind to the ion exchanger. The ionic strength should be relatively low to avoid damping of interaction between solute and ion exchanger. Buffer concentration in the range of 0.05 to 0.1 molar are recommended. Now, application of ion exchanges, ion exchange chromatography. The separation and purification of proteins, peptides, nucleic acid, polynucleotides and other charged molecules mainly because of its high resolving power and high capacity. It is used to separate DNA from cell extract, separation and removal of inorganic ions from aqueous mixture. Next one is partition chromatography. The differential distribution of solute between two immiscible liquid is the basis of separation of partition chromatography. It can be subdivided into liquid-liquid chromatography and bonded phase liquid chromatography. In liquid-liquid chromatography, the liquid stationary phase is attached to a supporting matrix by purely physical means. In bonded phase liquid chromatography, the stationary phase is covalently attached to the matrix. For example, a water stationary phase is supported by a cellulose, starch or silica matrix, all of which have the ability to physically bind as much as 50% water and remain free-flowing powders. Now, application. It is used to separate analytes that have low water solubility and those that are not amenable to reversed phase liquid chromatography. Coming to the adsorption chromatography, certain solid material, collectively known as adsorbents, have the ability to hold molecules at their surface. This adsorption process, which involves weak non-ionic attractive forces of the van der Waals and hydrogen bonding type, occur at specific adsorption sites. Silica is a typical adsorbent. It has silanol group on its surface which are slightly acidic and can interact with polar functional groups of the analyte or eluent. Other commonly used adsorbents such as alumina and carbon. Next one is affinity chromatography. It does not rely on differences in the physical properties of the analytes. Instead, it exploits the unique property of extremely specific biological interaction to achieve separation and purification. This technique requires that the material to be isolated is capable of binding reversibly to a specific ligand that is attached to an insoluble matrix. When a complex mixture containing the specific compound to be purified is added to the immobilized ligand, generally contained in a conventional chromatographic column, only that compound will bind to the ligand. All other compounds can therefore be washed away and the compound subsequently recovered by displacement from the ligands. Coming to the steps which are involved in it, first, choice of appropriate ligand. Second, immobilization of the ligand onto a support matrix, binding of molecules of interest with the ligands, removal of non-specifically bound molecules, elution of molecules of interest in purified form. Coming to the choice of ligands, factors to be considered for the choice of ligands are specificity, reversibility, stability, size, affinity. Now coming to the applications, many enzymes and other proteins including receptor proteins and immunoglobulins are purified. Messenger RNA is routinely isolated by selective hybridization. Immobilized single-stranded DNA can be used to isolate complementary RNA and DNA. Immobilized nucleotides are useful for the isolation of proteins involved in nucleic acid metabolism. Immunoaffinity chromatography. The use of antibodies as the immobilized ligand has been exploited in the isolation and purification of a range of proteins including membrane proteins of viral origin. Monoclonal antibodies may be linked to agarose matrices by the cyanogen-bromide technique. Protein binding to the immobilized antibody is achieved in neutral buffer solution containing moderate salt concentration. Ions or small molecules that increase the water soluble of non-polar substances such as thiocyanate, perchlorates, trifluoroacetate, or lowering the pH to about 3 can be done for elution. Next one is size exclusion chromatography. The separation of molecules on the basis of their molecular size and shape exploits the molecular sieve properties of a variety of porous material. Size exclusion chromatography includes gel permeation chromatography and gel filtration chromatography. Volume of microparticulate cross-linked copolymer generally of either styrene or divinyl benzene with a narrow range of pore size is in equilibrium with a suitable mobile phase for the analytes to be separated. Large analytes that are completely excluded from the pores will pass through the interstitial spaces between the particles and will appear first in the invert. The smaller analytes will be distributed between the mobile phase inside and outside the particles and will therefore pass through the column at a slower rate, hence appearing last in the invert. Gel filtration chromatography. Gel filtration chromatography can be used to separate compounds such as small molecules, proteins, polysaccharides, nucleic acid when in aqueous solution. When organic solvent is used as a mobile phase, the process is known as gel permeation chromatography. Application of it can be used for fractionation of molecules and complexes within a predetermined size range. Size analysis and determination. It is used for removal of large proteins and complexes. It can be used as buffer exchange, desalting, removal of small molecules such as nucleotides, primers, dyes and contaminants, assessment of sample purity, and finally separation of bound and unbound radioisotopes. Retention factor or RF value. In paper chromatography, the results are calculated by RF value, which is basically a distance traveled by a given component divided by the distance traveled by the solvent front. If RF value of a solution is 0, it means the solution remains in the stationary phase and thus it is immobile. If RF value is 1, then the solute has no affinity for the stationary phase and travels with the solvent front. Paper chromatography. Paper chromatography is an analytical technique for separating and identify both colored and colorless mixtures. In paper chromatography, the stationary phase is a uniform absorbent paper which is made up of cellulose and whose nature is nonpolar in the form of paper sheet, makes an ideal support medium where water is adsorbed to the cellulose fiber and forms the stationary hydrophilic phase. Of separation, mobile phase rises up by capillary action. Testing sample is concentrated as a minute spot at the bottom of the filter paper. After that, sample mixture gradually rises up with the mobile phase, which is a liquid. Compound in the mixture will be separated according to their ability of solubility. More polar substances will move slower and less polar substances will travel faster. Sometimes it is rather difficult to separate a complex mixture of a substance by a single run with one solvent system. In such cases, a second run is carried out by a different solvent system in a direction perpendicular to the first one. This is referred to as two-dimensional chromatography. Affinity chromatography. Affinity chromatography is essentially a sample purification technique used primarily for biological molecules such as proteins. In this method, separating a mixture of protein or nucleic acid by specific interaction of these molecules with a component known as ligand, which is immobilized on a support. For example, if a solution of a mixture of protein is passed through the column, one of the protein binds to the ligand based on specificity and high affinity, whereas other proteins in the solution wash through the column because they are not able to bind to the ligands. Now, principle of affinity chromatography. It is based on highly specific biological interaction between two molecules, such as interaction between enzyme and substrate, receptor and ligands, or antibody and antigen. Ion exchange chromatography. Ion exchange chromatography is a process by which a mixture of similar charged ions can be separated by using an ion exchange reagents, that is cation exchanger and anion exchanger, which exchanges ions according to the relative affinities. The principle behind ion exchange chromatography, it is based on the separation of molecules on the basis of their electrical charge. Size exclusion chromatography. Size exclusion chromatography is also known as gel filtration or gel permeation chromatography, which uses porous particles to separate molecules of different sizes. It is generally used to separate biological molecules and to determine molecular weights and molecular weight distribution of polymers. It is usually applied to large molecules or macromolecule complexes such as protein and industrial polymers. When an aqueous solution is used to transport the sample through the column, this technique is known as gel filtration chromatography. On the other hand, when an organic solvent is used as a mobile phase, the technique is known as gel permeation chromatography. Size of pores in beads determines the exclusion limit. What goes through the beads and what goes around the beads is used to transport the sample through the column. The technique is known as gel filtration. Principle is based on the separation of molecules on the basis of their molecular weights. Application of chromatography. The chromatographic technique is used for the separation of amino acid, proteins and carbohydrates. It is also used for the analysis of drugs, hormones, vitamins. It is also helpful for the qualitative and quantitative analysis of complex mixtures. The technique is also useful for the determination of molecular weight of proteins. Paper chromatography. Introduction. Paper chromatography is one of the types of chromatography procedures which runs on a piece of specialized paper, generally Whatman filter paper. It is a planar chromatography system in which a cellulose filter paper acts as a stationary phase on which the separation of compounds occur. It is the simplest and the commonest form of liquid-liquid chromatography, that is partition chromatography. Whatman filter paper or commercially prepared cellulose plates are used for chromatographic separations. What happens in paper chromatography? In paper chromatography, the stationary phase is a special quality paper which is known as chromatographic paper. Mobile phase is a solvent or a mixture of solvent. A solution of the mixture is spotted on a line which is about 2 cm above from the bottom of the paper, which is called original line or the base line, and then suspended in a chromatographic chamber which contains suitable solvent. Thereafter, the solvent rises up the paper by capillary action and flows over the spot. The paper selectively retains different components of a mixture according to their different partition in the two phases. The paper strip is so developed is now called as chromatogram. The spots of the separated colored compounds are visible at different heights from the position of the initial spot on the chromatogram. The spots of the separated colorless component may be observed either under ultraviolet light or by the use of an appropriate spraying agent. Now coming to the RF value. RF value is also known as retardation factor or the retention factor. It is the ratio of the distance travelled by the solvent front from the original line and the distance travelled by the solute from the original line. Principle of paper chromatography. The principle involved is partition chromatography, in which the substances are distributed or partitioned between the two liquid phases. One phase is the water, which is held in the pores of the filter paper used. Generally, the filter paper is composed of the cellulose, and the cellulose molecule contains moisture, and the other is the mobile phase, which moves over the paper. The compounds in the mixture get separated due to the difference in their affinity towards water, that is stationary phase, and the mobile phase solvent during the movement of the mobile phase under the capillary action of the pores in the paper. The principle can be adsorption chromatography between solid and the liquid phases, where in the stationary phase is the solid surface of the paper and the liquid phase is of the mobile phase. But most of the application of paper chromatography works on the principle of partition chromatography, that is partition between two liquid phases. Now, types or modes of paper chromatography. Based on the way the development of chromatogram on paper is done in procedure, we have broadly 5 types of chromatography. First one is ascending chromatography. As the name indicates, the chromatogram ascends. Here the development of the paper occurs due to the solvent movement or upward travel on the paper. The solvent reservoir is at the bottom of the beaker. The paper tip with sample spots just dip into the solvent at the bottom so that spots remain well above the solvent. Next type is descending chromatography. The development of the paper occurs due to solvent travel downwards on the paper. The solvent reservoir is at the top. The movement of the solvent is assisted by gravity besides the capillary action. Next one is the ascending descending mode. As the name suggests that it is a mixture of ascending as well as descending mode. Here the solvent first travels upwards, that is ascending, and then downwards on the paper, that is descending. Next mode is the radial mode. The solvent moves from the center, that is midpoint, towards the periphery of circular chromatography paper. The entire system is kept in a covered petri dish for the development of the chromatogram. The wick at the center of the paper dips into the mobile phase in a petri dish, by which the solvent drains on the paper and moves the sample readily to form the sample spots of different compounds in concentric rings. Next one is the two-dimensional chromatography. The chromatogram developed occurs in two directions at right angles. In this mode, the samples are spotted to one corner of the rectangular paper and allowed for first development. Then the paper is again immersed in the mobile phase at a right angle to the previous development for the second chromatogram. Hence, two-dimensional chromatography is obtained. The paper chromatography procedures involves the following experimental methods. First one is the selection of suitable type of development. This depends on the complexity of the mixture, solvent, paper, etc. But in general, ascending type or radial type chromatography are used as they are easy to perform, handle, less time consuming, and also give faster chromatogram. Next one is the selection of suitable filter paper. Filter paper is selected which is based on the pore size, the quality of the sample to be separated, and also the mode of development. Then preparation of sample. Preparation of sample involves the dissolution of the sample in a suitable solvent which is making mobile phase. The solvent used should be inert with the sample under analysis. Spotting of sample on the paper. Samples are to be spotted at the proper position on the paper, preferably using a capillary tube. Development of chromatogram. How the chromatogram is developed. Samples are spotted on the paper is subjected to the development by immersing it in the mobile phase. The mobile phase moves over the sample on the paper under the capillary action of paper. Finally, drying of the paper and detection of the compounds. Once the development of chromatogram is over, the paper is held carefully at the bottom to avoid touching the sample spots and dried using the air dryer. Sometimes the detecting solution is sprayed in the developed paper and dried to identify the samples which are present on the chromatogram spots. This is an application of paper chromatography. Paper chromatography is especially used for the separation of mixture having polar and non-polar compounds. It is used for the separation of amino acids. It is used to determine organic compounds, biochemicals in urine, etc. In the pharma sector, it is used for the determination of hormones, drugs, etc. Sometimes it is used for evaluation of inorganic compounds like salts, complexes, etc. To check the control of purity of pharmaceuticals, for the detection of adulterants, to detect the contaminants in foods and drinks, to study of ripening and fermentation of foods, for the detection of drugs and dopes in animals and humans, for the analysis of cosmetics, and finally for the analysis of the reaction mixture in biochemical labs.